Okay, so why is this important? Well, especially if you're going to medicine, remember that you can't really change your cells like the inside of every single of the billions of cells in your body. So what you can do is change the tonicity and change the composition of the fluids surrounding your cells. And an IV BAT or an intravenous solution is a example of one of these extracellular fluids. So IV fluids are basically used in medicine and especially in nursing to balance out intra and extracellular fluids. So the great thing is that you can actually move water in and out of the cells by changing whether the solution surrounding a cell is isotonic, hypertonic, or hypotonic. So how do you do that? Well, osmosis. And this is what we see here. So here we have a normal brain, and over here we have a brain with something we call cerebral edema. So cerebrum refers to this skull area here and the brain. So, but what we have with the cerebral edema, well, okay, this is like an X-ray, and in general, when you have an X-ray, something that the whiter and brighter something appears, the more dense it is. The darker it is, the less dense it is. So what we actually have here is a lot of fluid surrounding this. This is why it appears kind of darker because, again, compared to the bones that make up your skull, which is very dense, which is why they appear white. And what we're seeing here is actually a transverse section. So if you took someone's head and took a slice here and looked at it, this is what you would see, something similar to this. Now this gray, like kind of medium gray color is your brain matter. But over here, you notice that this part over here is darker than this corresponding part on the normal brain. So what's that implying is that there's a lot of fluid accumulating in the brain. And you also notice that the folds here, well, you actually have something called sulci and gyri in your brain. So all these folds and bumps and grooves. Notice that there's so much, this brain is so waterlogged, you don't really see these folds because, again, this brain is really, really swollen. This is a bad example of being swollen. So what we see with the edema brain, another example, well, let's look at just the brain itself. Again, your brain should have all these folds and grooves, but in this very waterlogged brain with edema, you notice that all these little folds are kind of swollen and these grooves are also shallower. But why is this a critical case to address? Well, the thing is that your brain is not seen in this picture. Your brain has many blood vessels. So what happens if you're like, say, well, the buses and public transports, and if you're being in an area with like a subway, they're pretty empty now. Well, I think New York is actually back. But the thing is that what happens if too many people are on a bus or a subway? Well, you start to push yourself toward the air. You start to get very cramped and suffocated, right? So this is what happens to your blood vessels if you have cerebral edema. You have all these blood vessels in your brain, and if your brain is waterlogged, it's going to start to pinch off those blood vessels. And your brain needs blood because it needs a lot of oxygen. It needs a lot of nutrients as well. So how can we reduce the swelling? Well, we can reduce the swelling using osmosis. So what we can do is use osmosis. We have too much water in the brain, in the cells and the tissues. Let's use osmosis to draw out fluids from that area. So how did we do that in the previous examples? Well, we did place it into a different type of tonicity. So we can do something like a mannitol, IV, or oral glycerol. These have very high concentrations of solutes. Remember that water is drawn to the fluid that has more solute. So if you give a very high concentration of mannitol or oral glycerol, it is going to be hypertonic. A hypertonic extracellular fluid is going to draw water out of cells, and this is how you can reduce that swelling and waterlogging of the brain. You can give a hypertonic solution to draw water from the brain so that it relieves the pressure on all those blood vessels that supply the brain. Okay. Next top hat question. So your patient is suffering from dehydration and hypernatremia. So that means they have abnormally high blood sodium, too much blood sodium. So if say you only have these IV solutions available, which of the following would be best to treat their dehydration? And yeah, I know I'm giving 2 lectures on osmosis, but this is why osmosis is very important. Okay, let's see how people did. So most of you said hypotonic saline, and most of you are correct. So again, what we're having is too little water. So you wouldn't want to give hypertonic. Why? Because hypertonic is going to suck out more water from the cells and dehydrate your patient even more. So A and D are definitely stuff you don't want to give to this patient who's already dehydrated. Now, isotonic versus hypotonic. Actually, a case could be made for a isotonic, but this is where you have to kind of take in that hypernatremia part. Hypernatremia means they have too much sodium already, so you want to get water into their body and into their cells, but you don't want to add more solutes and more sodium. So the better solution between the B and C would be C, because it has less solutes than that isotonic solution. And actually, don't worry, this is like maybe borderline too difficult for this class, but I'm just showing you a practical example because this is kind of like almost something you might see on the NCLEX or a simplified version of what you might see for med school. So don't worry if you didn't get this one right, but if you got it right, kudos to you.